Aldehydes and ketones are unable to hydrogen bond with themselves, but they are able to hydrogen bond with water, and they have strong dipoles. This means that they typically have lower boiling points and lower solubility in water than alcohols of the same length, yet higher than ethers, which have much weaker dipoles. Back in chapter 14, we talked about how primary alcohols could be oxidized to aldehydes or to carboxylic acids, and we also talked about how we could add hydrogen, H2, to an aldehyde and end up with a primary alcohol. Primary alcohols, aldehydes, and carboxylic acids can be converted back and forth by redox reactions. Remember, a redox reaction changes the number of electrons assigned to an atom. Carbons are always more oxidized, that is, having fewer electrons assigned to them, when they're bound to more oxygens, since the oxygen will hog all those electrons. So a carboxylic acid will be more oxidized than an aldehyde, and an aldehyde would be more oxidized than a primary alcohol. Is a ketone or a secondary alcohol more oxidized based on this mode of looking at things? The main change that we introduce in this chapter will focus on how aldehydes can go directly to carboxylic acids, while ketones cannot go to anything more oxidized. Oxidation problems for aldehydes and ketones should look fairly familiar. We'll have a molecule and oxidation conditions, mild oxidation, strong oxidation, or KMnO4 or chromium-based reagents, which are also strong oxidants. In each case, the functional group will change, and it will change predictably. Let's start by identifying which functional groups are actually reacting as either primary, secondary, or tertiary alcohols, or aldehydes, or ketones. The top molecule is a primary alcohol. The next molecule is a secondary alcohol. The third molecule is an aldehyde. The fourth molecule is a primary alcohol. And the fifth molecule is a ketone. Once we know what type of molecule is reacting, we can use that to predict the product. So if we look at the top molecule again, it's a primary alcohol and it has mild oxidizing conditions. We know that that yields an aldehyde. A secondary alcohol under strong oxidizing conditions like KMnO4 will yield a ketone. An aldehyde with any oxidant will yield a carboxylic acid. A primary alcohol under strong oxidizing conditions like anything with chromium, chromium 6 oxide or CrO3 in this case, will yield a carboxylic acid. And finally, no matter how strong the oxidant, a ketone will never react. It will have no reaction. Once we've identified the type of product our compound is going to be, we're really just drawing the same carbon skeletons and swapping out the functional group. So for the first molecule, we draw the same 4 carbons, and where there was an alcohol, then we put an aldehyde. Pause and try it yourself before you check your work. A second class of reactions reacts an aldehyde or ketone with an alcohol. This is really important as a reaction because all carbohydrates have an aldehyde or ketone, and they also have several alcohols on the same molecule. So they will actually self-react and form a different form. Even though we might see D-glucose listed like this in your biology text, you'll see it change to this, which is also D-glucose pretty soon afterwards. Both are true. Glucose reacts with itself to form a type of functional group known as a hemiacetal. Similarly, fructose is typically drawn like the molecule on the left in textbooks, and then it's switched to this. Hemiacetals are made from reacting an aldehyde or a ketone with an alcohol. In the reaction shown, a ketone and an alcohol are reacting, but it could just as easily have been an aldehyde. The hemiacetal that forms has an OH, an alcohol that is, and an OR, an ether. The OH and OR need to be on the same carbon for this molecule to be considered a hemiacetal. The hemiacetal will always consist of portions that correspond to both the aldehyde or ketone and to the original alcohol. In this molecule, the 5-carbon chain with the ketone on the second position still will remain a 5-carbon chain, now with a hemiacetal on the second position. So again, we're not changing the carbon skeleton, we're just changing to a new functional group at that location. The alcohol that's added across the double bond adds with a more negative O and the carbons attached to it, this section in blue, going to the carbonyl carbon. So we see that attached to the carbon that was double bond to oxygen before. The H, which is more positive, will add to the carbonyl oxygen, which is more negative, creating the alcohol in the hemiacetal. I highly recommend that you color code the reaction yourself in your notes so you can see which group goes where. The formation of a hemiacetal is reversible. So if a ketone and an alcohol form a hemiacetal, they can break back apart again as well. That's why we see the arrows going in both directions in this reaction. The reaction is typically facilitated by water, which I promise you your body has. So you're always shifting back and forth between these two forms. If there is sufficient alcohol, that's enough alcohol, a hemiacetal can further react to form an acetal, which we can see highlighted there. The acetal is characterized by two ethers that share the same carbon. Again, the ethers have to be on the same carbon for it to be an acetal. Otherwise, it's just two ethers. This reaction is much more permanent and corresponds biologically to the formation of di- and polysaccharides from monosaccharides, our simplest carbohydrates. It can be reversed in strong acid or base, or in our bodies by enzymes. Just like before, we can track pieces of this molecule, or of all of the molecules. Here we have all the same colorings as before for our hemiacetal. So we can see the yellow is the portion that came from the aldehyde or ketone. In this case, it's a ketone. We can also see the portion in blue that came from the alcohol. Again, we will add the alcohol in two parts. The majority of the hemiacetal is maintained in the acetal, with the second alcohol portion replacing the OH on the molecule. The OH from the hemiacetal gets the hydrogen from the alcohol and is removed from the water as molecule as water. We don't need to draw this water, but it's sometimes helpful to see where the atoms went. So by adding a second alcohol on, we've actually kicked out the oxygen that was originally on there. In the acetal, we can still clearly identify three groups: two ethers that correspond to the original alcohols, and a carbon chain that's bound to both ethers that corresponds to either an aldehyde or a ketone. The aldehyde or ketone part always has a carbon that's connected to two oxygens, no matter what form it is, aldehyde or ketone, hemiacetal or acetal. So we start out with a double bond to oxygen, then we have a bond to an alcohol oxygen and an ether oxygen in a hemiacetal, and by the time we get to an acetal, we have two bonds to ether oxygens. But in every case, that same carbon will always be bound twice to oxygen. Before we get into drawing products, it's important to be able to identify which molecules are hemiacetals or acetals. That'll help us to actually transform these correctly, and which are neither. Remember, hemiacetals will have an alcohol and an ether on the same carbon, and acetals will have two ethers that are on the same carbon. From the top left, we have a molecule that's neither hemiacetal nor acetal, since the alcohol and ether on this molecule do not share a carbon. So this is actually just an alcohol and ether, has completely different reactivity. The next molecule is a hemiacetal, because the OH and OR, the alcohol and ether, share a carbon. On the top right, we have another hemiacetal. The OH and OR again share a carbon. In the bottom left, we have another hemiacetal. The OH and OR share a carbon, and even though it's in a ring, it's still a hemiacetal. And finally, we have an acetal, where we have the carbon that's right here has two ORs attached to it, making it an acetal. The bottom two structures correspond more or less to the basic structure of a monosaccharide and a disaccharide in our bodies. In each of these reactions, we're trying to predict what the product of each reaction looks like. For these examples, just for the examples we're going over right now, we'll use the same alcohol, methanol, which is abbreviated MEOH in each case. That way we can get used to just seeing that transformation as a functional group transformation. In the first reaction, we'll create the hemiacetal version of the molecule, again where we have an OH and an OR, an alcohol and an ether. And the second reaction, because we're adding more alcohol, we'll create the acetal version. So in the top row, we have a hemiacetal with the same cyclohexane ring, but now we also have an alcohol and a methyl ether at the carbon that used to be a ketone after it reacts with methyl alcohol. So the methyl alcohol makes the methyl ether. If we had an ethyl alcohol, it would make the ethyl ether. If we react again, we have two methyl ethers at that carbon. Now pause and see if you can identify the hemiacetal, that's the left column, and the acetal, the right column, that are formed from adding methanol to each molecule. We'll always see the carbon chains stay the same, so we continue to have the two carbons leading up to the hemiacetal or acetal side of the reaction in the next step. So these two carbons right here are still the two carbons that show up here. And the hemiacetal will have an alcohol and a methyl ether. The acetal will just have two methyl ethers from the two methyl alcohols. In the bottom row, we have benzaldehyde, which reacts similarly, first forming a hemiacetal with an alcohol and methyl ether where the aldehyde was, and upon reacting with a second alcohol, it will form an acetal with two methyl ethers. We can also have these reactions happen intramolecularly. That means within the same molecule. Intra is within the same, and inter is between different ones. This is a little bit hard to see, but way more important because this is what happens in carbohydrates. Just as before, the alcohol oxygen will form a new bond to the carbonyl carbon, and the H will go to the carbonyl oxygen. It's easiest to look at it in the first place like this, where we just draw this bond straight from the oxygen to the carbonyl carbon, and we don't worry about changing the arrangement of molecules. Notice that the carbons that were connected to the alcohol stay connected to it, now forming a ring. You probably won't see it written like that though, because one bond is way longer than the others. But we know that there are 5 carbons and an oxygen in a ring, so 6 total things, so we might see it drawn like this instead. Hopefully this looks really similar to the carbohydrate structures we saw before, because this is exactly what happens to the carbohydrates. Just like we saw versions of alcohols and ethers with the oxygen swap for sulfur, there are versions of aldehydes and ketones with the oxygen swap for sulfur called thiols and thione. These aren't very stable because the sulfur can easily be oxidized. We care a lot more about sulfoxides, which have the carbonyl carbon swapped out for sulfur. These can be produced by oxidizing thioethers and are very stable molecules similar in property to ketones. Dimethyl sulfoxide, or DMSO, is the only one that we'll see with any sort of frequency, so you should memorize the name and structure of DMSO. It actually shows up a lot in biological solutions.